All right, let's go ahead and get started here. First of all, I hope you enjoyed your Friday off. So it's been a while since we have met together. Quick announcements before we get started on lecture today, though. Advising day is tomorrow, and there is a lot going on. All right, so I would encourage you to check out a few things here. One of them is, I know this is Bio 201 exam and study tips. Open to all students. If you have any questions about studying or tips in general, there'll be some tutors available for you in Nobel here at 7:30. That got me confused. It said November 7th at 7:30. So at 7:30, there'll be tutors available just to ask some general questions, or if you have one-on-one questions as well. If you are considering majoring in something in science, which you're in this class, I'm going to assume all of you are in some aspect of science here. But if you're considering majoring anything in sciences and Nobel, like geography, geology, chemistry, biology, there's a major fair happening tomorrow. You're in Nobel. It's by the big globe in the North Atrium over there. So another thing that you can attend. And then finally, if you have any questions about registration, there's going to be an advising day headquarters in the Heritage Room in the Campus Center. So if you want to do any drop-in advising, if you have questions about Mike Gustavus, anything happening, you have an interesting situation that you don't know how to handle, there will be people there to talk about advising. So I would encourage you to look at all of those fantastic resources. All right, let's move into lecture. We are a little behind where the schedule says that we should be at, all right, but we'll catch up in the next day or two here. But we did end our last lecture on this slide here, right, where I asked you to draw this cell in metaphase of meiosis 1 and meiosis 2. Right? And I asked you what the total number of chromosomes were for that second cell. Right? We had started going through the answers for this top one here, right? And I have made them a little bit neater on this slide. So if you remember right, 2n equals 6. That means 6 total chromosomes in a diploid cell, right, which would mean 3 homologous pairs. You can see we have these 3 homologous pairs. They are each different sizes, so that's a good giveaway, right? In metaphase of meiosis 1, you should have drawn them as tetrads lining up on that metaphase plate, right? Now, something I couldn't draw very easily digitally was you could have also drawn a little bit of crossing over that would have already occurred in prophase, right? So you'd still see it in metaphase, even though it already happened, right? And then if we move this cell through metaphase of meiosis 2, right, like instead of having these tetrads line up, now we are having the individual chromosomes line up, and eventually those sister chromatids would split, right? I drew 2 here because if we split this, the tetrads in half here, we would end up with the next arrangement something similar to this one here, all right? We also did not get to this next question here. So if I had a cell that was 4n equals 8, what is the total number of chromosomes in this cell? I hear the whispers. 8, yes, 8 is the total number. We see 4n equals 8. How many different types of chromosomes are there? 4. Yes. So normally we see haploid or diploid cells, right? This is a weird one. This is a tetraploid, which means instead of a homologous pair, they have 4 chromosomes that all contain the same type of genetic information, right? So these would be in groups of 4, right? And maybe I answered that wrong. I see why some of you were saying 2 there. Because yes, there would be 2 types of genetic material present, right, in groups of 4, right? So if I were to draw this cell, right, it would look something similar to up here, right? I drew it before replication and after replication. each of them would be correct in the number of chromosomes that they have and who they're homologous to. Any questions about this review? Yes? It would be. Yeah. I can see where you said that. Yep. And how many of each type? 4. Yep. Any other questions, things I can clarify? All right. So we're going to move on to a little bit more about chromosomes, right? And so chromosomes, they can vary. They can vary in their structure and in their number. And we've talked a little bit about this with diploid and haploid and tetraploid cells, right? And if we have variation in chromosomes, depending on how that variation occurs, right, we can get different things going on in our organisms, right? We could get a human disease, right? So sometimes if our chromosomes don't split right, or there's some chromosomal structure difference going on, we could get a disease. In other species, particularly those that are not mammals, right, we could end up getting an evolution of a new species down the road if we have difference in chromosome structure and number, right? Big picture on this slide here, right? If we're talking about members of the same species, right, difference in number and structure of chromosomes is rare, right? If it's the same species, so say all of humans or all of cats or all of dogs, right? Between humans, right, differences in the number of chromosomes you have and their structure is pretty rare. But if we look between species, right, so now we're comparing humans to cats, to dogs, to lizards, to plants, right? That's actually pretty normal to see variation. And we're going to go through a couple examples here. So we can change our chromosome structure, right? And this can happen in a variety of ways, right? I put all of the ways up on the screen here. But you can see we could simply delete part of our chromosome, right? That's an abnormality that can happen. We could duplicate part of the chromosome. We could flip the order of the chromosome around. Right? We still have that genetic material, it's just flipped in the order on our chromosome. If we look at two different chromosomes, right, we could take this chromosome, that's like blue and yellow and black there, and put it on top of the bottom part of this red chromosome. Right? That would be something called translocation. We could have reciprocal translocation, where not only are we putting part of a chromosome on the other chromosome, but vice versa is also happening. So we're modifying two different chromosomes as well. Right? There are some unique things that can happen. Right? And it only depends on what genes are being altered here. Right? Sometimes if they're important genes to cell function, this could be lethal. Sometimes it's not lethal, but it could lead to genetic diseases as this gets passed down through generations, right? Completely depends on the genes that these are happening to. All right, so that's variation in structure. But if we talk about variation in number, this is really common between species. Right? So like I said, if we have different number of chromosomes from a dog or a frog, right? That's common, right? So humans, we have 46 chromosomes, which we know are in 23 pairs. Dogs have 78. Fruit flies only have 8. Potatoes have 48. Yeast have 32. Right? There is variation in biology. Right? So that is very, very common. Right? So if we look at differences in chromosome number, there's a couple different ways that it can vary, besides just having a different number from another species. So now if we're talking about the same species, right, these are two variations that can occur. Right? The first is aneuploidy. Right? This would be a variation in the number of individual chromosome types. So for example, trisomy here is where instead of we having like a pair of chromosomes, instead of us having a pair of chromosomes, we might have 3 of a chromosome type. Right? This is actually pretty common with those individuals with Down syndrome. Right? Instead of having 2 of our chromosomes, chromosome number 21 specifically, right? They have 3. They have an extra chromosome in there, right? So that would be tricity. We're adding in an additional chromosome to one of our sets, right? We're not adding in an additional chromosome to every single one of those 23 pairs, just one of those pairs, or a couple of those pairs, right? Monosomy would be the opposite, right? Instead of having 2 chromosomes in a diploid or in a pair here, we would only have 1, right? So a common example of this is Turner syndrome, right? So these are looking at the sex chromosomes here. So we have 2 X chromosomes, which would be normal to have, right? But in Turner syndrome, individuals might be missing one of those X's, so they only have 1 chromosome instead of 2. Right? So that's aneuploid. We're changing the number of chromosomes for chromosome types. Right? So instead of a homologous pair, we might have 3 of a kind, or we might only have 1. Now, polyploidy, on the other hand, is something that we have talked about. Right? We have seen the terms haploid and diploid and triploid. Right? So in polyploidy, we are having variations in the number of chromosome sets. Right? So the entire genome as a whole has been duplicated in some manner. All right, so diploid, we know we have 2 homologous pairs or 2 chromosome sets. Triploid, we would have 3 chromosome sets. Right? We can get tetraploids with 4, octoploids. Right? There's even a plant species, super, super tiny plant species, that is 96ploid. Right? So they only have 15 sets of chromosomes, but each set has 96 chromosomes. That's crazy, right? That's like 14, that is 1,400 chromosomes, right? In that tiny little plant species. All right, so let's back up to aneuploidy. So aneuploidy, we're changing the number of chromosome types for a particular set. And if we're talking about humans, this is typically lethal, right? Not always, but most commonly it's going to be something that's very lethal, right? And so if we have, if you look at some statistics here, about 5 to 10% of all fertilized human eggs result in some sort of chromosomal abnormality. We're talking about individuals who experience a spontaneous abortion. Right? About 50% of the time, that is due to an alteration in chromosome number. Right? So it's typically lethal, but not always. There are some exceptions. Right? So individuals with some of these variations can survive. Right? So if we have trisomy on our 13th pair, our 18th pair, or our 21st pair, which is what we looked at before, Down syndrome here, those individuals can survive, right? As well as some abnormalities in our sex chromosomes. We talked about Turner syndrome on that last slide, right? So usually lethal, not always, right? How does this happen? Well, for this to happen, we have to have non-disjunction occurring during meiosis. Right? Formation of gametes going through meiosis here, and either in meiosis 1 or meiosis 2, those chromosomes are not going to split apart correctly. So if we look at the example over here, we have non-disjunction in meiosis 1 here. This would be occurring during anaphase. And we're splitting apart those tetrads, and we see that we have 1 tetrad that is not being split apart. So essentially we have an excess of genetic material going to one side of the cell versus not enough genetic material going to the other side. And if we follow this cell through meiosis, we see the resulting gametes all have abnormalities in cell number. They either have an extra chromosome or they are lacking a chromosome. Yes. random, but I'm not sure the precise mechanism behind it. I'm going to assume it's something where the mitotic spindle is not connected to the kinetic wheel, or there's some interference there, but I don't know specifics off the top of my head. Yeah, good question. Any other questions? All right, so our example on the other side of this image here, pretty much the same, except now it's happening in meiosis II. So this is the failure of our sister chromatids to separate apart, right? And we see the resulting gametes, we have 2 of them here that have an abnormal number of chromosomes, right? So non-disjunction, it occurs, right? Usually lethal, not always. All right, but if we talk about polyploidy in humans, this is always going to be lethal, right? But like everything in biology, there's exceptions, right? So actually in our liver and in placenta, we actually do see polyploidy. And this is an area of active research, right? So I tried to figure out why, right? I dug into some of the literature. And what I could find is in the liver, right, where we have polyploidy, they're thinking, the working hypothesis is that we have extra chromosomes containing genes relating to tumor suppression. So we're trying to suppress any tumors from forming in the liver. Yes? Very close. Yes, so difference between ploidy and polyploidy. Ploidy is just talking about chromosome sets in general, broadly. Right? Ploidy levels could be diploid, haploid, tetraploid. Right? Polyploidy is specifically meaning to more than 2 in a set. So polyploidy would be something like 3 or 4 or 5 in a set. So it's a similar terminology, but slightly different. Does that make sense? All right. Yes? So when you say apply that term to all of them. They have more than 2 in a set. It is a polyploid organism. Your question? So our liver and placenta cells have a different yeah. Yeah. But if that would have happened to all of us, we would have. Not Down syndrome. If it would happen to every cell in our body, we wouldn't survive. So why do we have polyploidy in some of these tissues? Some ongoing research. It's interesting. Yeah, we know it exists in placenta. We don't really know why yet. But like I said, if this were to happen to all of our cells, it's going to be lethal, right? So if in a fetus that is developing is triploid, right? So they have 3 in each chromosome set. It can be live born, but it will die shortly after. Right? If it's a tetraploid, right? It could start to form, but it's not going to survive very far into the pregnancy. Right? So polyploidy in an entire individual in humans, very lethal. But that's not saying that polyploid everywhere is lethal. All right, in humans it's lethal. In most mammals it's lethal. But there are a whole host of organisms out there that this is very common. Right? And so this image up here is just showing you a snapshot of those. Many plants are polyploids, fish, amphibians, yeast, right? They have polyploidy to them, and it's very common, and it's normal, and they can function as usual. Right? In fact, polyploidy is very, very common in plants. All right? It would be almost unusual at this point to find a plant that's only haploid or diploid, but instead has some sort of polyploidy in their genome. All right? And so we think that this polyploidy have led to new species arising in plants. In fact, about 70% of vascular plants are polyploids at this point, and we think they were recently evolved. Plants that are polyploid polyploids can tolerate harsher conditions, right? And that can allow them to self-pollinate as well, right? So plants actually can have an advantage to being a polyploid, right? So you kind of have to think about the human side and everything else out there, which is way more all-encompassing than just humans, all right? So I did want to point this out that polyploidy is very common. All right, let's take a step back and do some group review here. I want you to look at these karyotypes and see if you can figure out, is it an example of aneuploidy or polyploidy? All right, I'll give you a couple minutes. I'm going to keep passing back some papers here. If you have questions, let me know. All right, so let's look at this image at the bottom here. What is this an example of? Aneuploidy, yeah. What chromosome sets are the oddballs here? 14, yes. There's a lot. Oh, I missed the X when I circled it. Yes, this is an example of an individual where there's a lot of aneuploidy going on here. All right, so we see aneuploidy in multiple sets, including X here, which is one that I did not catch when I was circling all the different variations. What about the one next to it here? What is this an example of? Yeah, polyploidy. Ooh. I did it out of order here. But polyploidy here, right? Because we have an entire genome, right, where every set has the same number, right? But it's a tetraploid because there's 4 in each set. Yes? Oh, did you put the, So, an example of being a polyploid, but there's an aneuploidy within this polyploid. So, that's a great example of being both, right? So if we're looking at this tetraploid here, I'm going to assume this came from a human chromosome, right? Now, not a human that survived, because we know this is lethal, right? But because we have 23 sets, right? But we see they were not only polyploid, right? But there was some aneuploidy in here that I didn't even catch, so very good eye, right? It's just some of them have 3 instead of 4. What about this top one here? I hear the whispers. Aneuploidy, yes. We see aneuploidy in our sex chromosomes there, right? We only have 1 chromosome in our sex chromosome instead of 2. Whether that second one should have been an X or a Y, I'm unsure, but it's definitely missing one. I don't know. I would have to do some research. Yeah. I'm just assuming it's humans because they're the most studied organism, which is why I would bet this was from a human organism, but it could have been from another organism that had 23. If anyone does a quick research, you can maybe figure it out. You can let me know. Yeah, good question. Do we have any other questions? All right. So we're going to move on to what was originally slated to be talked about today. We're only going to get through a little bit of it before we get to that content check at the end of lecture here. We're going to move on to gene expression. Right? We talked a lot about chromosomes and cell division, right? But now we're going to talk about how we take that information in our chromosome, right, and translate it into a trait that shows up, right? So we'll eventually talk about transcription and translation, but I wanted to start with a review. I know we've been talking about chromosomes, but chromosome terminology is confusing, right? Because there are several things that can be the same, right? They could look different but have the same name. So I want to make sure we are all starting off on the same page here. So if we look at our chromosomes, right? This is a singular chromosome, as well as this one is a singular chromosome, right? This one might be a chromosome from dad, whereas this one is from mom. They carry the same type of genetic material, right? So they would be a homologous pair, right? So they're carrying the same genes on either chromosome here. If we look at them when they're duplicated, they would look something in this format here where we see that typical X shape, right? But this is only after DNA replication when the cell prepares to divide, right? Most of the time, they would just be individually without that replicated half, right? But when they are replicated, this is a singular chromosome, and that is a singular chromosome. Right? So that's where it can get kind of confusing. Chromosomes can look different and still be called chromosomes. Right? It's because they contain the same genetic information. It's just duplicated when it's preparing to divide. And because they're containing the same genetic information, still a homologous pair. Right? So I'm going to go back to when they're just singular here. Right? Because this is how we see cells most of the time. Right? Most of the time, our cells are not actively dividing. They're in interphase. Right? So we have our homologous pair here. And so on each pair, we would have traits for genes. This one is specifically pointing out a gene for eye color. Right? So a gene is a sequence of DNA that will eventually be transcribed into RNA and then seen as a functional product, right? Which is where we're going to head with this lecture on Wednesday here, right? So a gene is that section of DNA that will eventually be translated into proteins and will have something that is functional, right? Now specifically for this gene, that would translate into eye color, right? Now, we get one from dad, one from mom. We know they're a homologous pair. They contain the same type of genetic information, but there can be variations, right? There can be different alleles, right? So an allele is basically a variation of a gene, right? It's one of at least 2 versions of a gene, right? We could have 3 versions. In our example here, right, coming from Dad, we have our allele for brown eyes. Coming from Mom, we have the allele for blue eyes. Right? It's the same type of genetic information, but different variants of it. Right? Now this one specifically talks about alleles being dominant and recessive, which is something that you will go over in lab this week, and we'll talk more about next week. Right? But if this allele is dominant, it means it's going to overshadow any recessive allele. And so this individual, when it has this set of chromosomes here, this homologous pair, will show eye color as being brown because it's the dominant allele. Even though they have DNA for blue eyes, it's not the one being expressed. Right? Which leads us to our next slide of gene expression. Right? Any questions on chromosome terminology though? All right. So every single one of our cells has the same genetic information, right? We contain all of our DNA in all of our cells, right? So why do cells look so different and act so different, right? Red blood cell carrying oxygen through our bloodstream looks very different than these muscle cells that will contract as we flex our muscles, right? Very different from a bone cell or a nerve cell, very, very different. But they all contain this exact same copy of DNA, right? Well, they're different because of gene expression, right? So gene expression is the process where this information is either encoded or not. So basically, in simple terms, we can say the gene is being turned on or off, right? The gene is being expressed, or the gene is being turned on, or the gene is not being expressed, or the gene is off. Right? So by expressing different genes in different cells, we can get variation in how our cells look and how our cells act. All right, and we can look at this gene function in 2 different levels here, and these levels very much coincide with each other. So we can look at molecular function, right? So what is the function of the protein product of the gene? And the trait. So what is that trait that gene is conferring, right? So let's look at an example here. So on the bottom here, we have a chromosome with the gene for flower color, right? So we're looking at a plant chromosome here, right? We see this is our DNA sequence for this gene. This DNA sequence, right, through transcription and translation, will turn into a protein, right? I shouldn't say turn into, that gives you the idea that this sequence is rearranging to become a protein, but it's going to be translated into a protein. Right? This protein's function is to create a purple pigment, right? So that's its molecular function, and the trait is that we get purple flowers, right? So they're intertwined with each other. Now this is a very obvious example, right? So the protein that's making the pigments is showing up as something that we can visibly see on this plant. But a lot of times these things are not as obvious, right? So if we think about different proteins that can be made, they can be proteins that might be an enzyme, right? We're not going to see the enzyme, but the enzyme is going to have a function or a trait that goes along with it. It might be something like transport, so the hemoglobin here, right? Transporting oxygen, right? That's a protein, right? That was made from a gene, right? It's not something that's as obvious to us. We can't see it with our naked eye like these purple flowers, right? But still has a trait associated to it, right? So all of this is saying we are leading up to one of the most important pieces of biology is the central dogma. All right? The central dogma is how we go from DNA, right, transcribed into a strand of RNA, and then translated into a protein that confers a trait. And if you remember right, our proteins are made up of those 20 amino acids. Right? And it's this transcription and translation that's part of gene expression. It's what's turning these genes on or off. So not every gene in every cell will always be expressed, only a subset. Any questions? All right, we're going to go over transcription and translation on Monday. There is no homework, not Monday, on Wednesday. I don't know why I'm thinking it's Friday today. Wouldn't that be nice? We're going to go over it on Wednesday here. There's no homework, but keep in mind there is a content check due on Friday. I will post that shortly. I didn't want to post it too soon because I didn't know how much material we would be able to get through. And then the exam is on a Monday, so keep that in mind as well. And then Monday, November 20th. All right, you have that content check, and you've been waiting for that question, that in-class question. I want you to draw. I know, I love having you draw. It's that muscle memory, committing it to paper instead of just typing out the words. I want you to draw the steps of meiosis 1. Right? You only have to do meiosis 1. You don't have to go on to meiosis 2 of a diploid cell with 3 pairs of chromosomes. Right? This should be review from lab last week, so it's a good chance to test yourself. And yes, I did bring a stapler this time, so when you are done, and if you have anything to staple, I will put it right up on here. Yes. Yeah, so the cell cycle. What is the cell cycle? D1, X, 2, and so you're not drawing a cell, you're just drawing the cell. You're drawing the producing. It's a diploid cell. It's a diploid. Yeah. Okay. Yes. Yes. Right. You're right there.
So I don't know what the reason is, but I had to ask you my parents they wanted to go on a trip.
Okay.
And I saw that we have an exam at 8:30.
Yes.
Is
Yes.
there any chance I would be able to take it the Friday before? Like I don't know like what the reason is.
Yeah, it is. It's hard.
I know that's why I wanted to use this
Right.
Or even Saturday. I don't know.
Can you send me an email?
Yeah.
It's likely we can work something out. I just have to double check everything to make sure everything's
Alright.
okay with policy.
I saw that Friday. I thought this would be like a study day or
Okay.
I think or something. But I just
Yeah, it might have been
I
reading
think
day.
so,
Yeah.
something like that. So,
Mhm.
but I just wanted to make sure.
Okay.
I wanted to conversation Friday.
Yeah. No, thanks for letting me know well in advance. That is helpful. Send me an email so it's on my radar. Because I will likely forget about this in a couple hours. We can probably work something out.
Okay. Alright.
It's tricky because I'm not going to be on campus that Friday and Saturday.
Okay.
But I'll see what other sections are doing, and you might sit in with another
Well, otherwise,
lecture section and take it.
if that works, otherwise, I mean, if you just stop Friday, I think that my Thursday schedule is super late.
Yeah.
So, I mean,
We
whatever.
will
works,
figure
I'll
something
see.
out. Yeah. Yeah.
Alright, thank you. And then I can turn that conference chat on.
Yes.
Alright.
I'll be right there. We had a question behind you. Just my own sister. You should start with ProPhase 1. That's for... Yes, that would be the cell center. Right, Hannah, we had a question. Yeah, I mean, a tetrad, it's just the formation of it close to each other.
So would it just be like those two?
Yeah, yeah. Yes, yes. They're just close enough to each other where they can go through crossing. Yep. And so, I like that you have ProPhase and I wouldn't technically be wrong, but in the cell, they would be scattered all about in different modifications, which is why that kind of ProPhase, you would have the tetrads coming together. So, each other. You don't have colors to indicate that, you could just say crossing over, occurs here. It's like a double row. Hold on. Let me go to a different slide in the PowerPoint here, and I think it'll click. So when we did this review up here, right, a metaphase of meiosis one, we had like two double rows of chromosomes, so it's like that. Yeah, yep. And then when they split, it's not, it's just a truncate, it's just yeah, okay, yep. Yeah, it feels so bad for you, you've been sick for so long. It's coming out in like this. Oh. I'm glad you have your handy tissue.
Thank you. I'll see you Wednesday.
Yes. Yeah. That's fine. Just drop it in my mailbox. Yep. So there's a great one. We have about a minute left here. So if you don't finish that in time, you can drop it off in my mailbox for Wednesday. Yeah.
And the other part is came in online.
Perfect.
Thank you.
Does this seem right?
Yeah, and I like how you drew the crossing over had a curve. Yep.
I'm just confused with that.
No, because remember, even if the chromosome is duplicated in the X shape, it's still a chromosome. So let's see. So we have one of the tetrad crossing over a curve. Single fully packed. Oh, I mean, you drew it correctly, right? So like these two purple ones would technically be joined in the middle by a centimeter.
Oh, so then
That would make it one.
Okay.
Yep.
thank you.
Yeah.
but crossing over, it doesn't look like it, but where it happens in prophase, right?
Mhm. Yep.
And
Yep.
then by the time they're there, they're already like.
Yeah.
Okay.
I like how you drew that crossing over courier. Just remember it
It
happens
is
in
there.
prophase. Yep.
Okay.
Yep.
So just color it differently.
Yeah. But you said it happened in
Yeah,
that
like,
phase,
I-
so I understand.
Oh yeah, I was like,
Yeah. Says
I
it was
know
due
it's
last
not.
Friday, huh?
I know it's not due then, so I wasn't too worried.
I hope our class visitor had a good visit.
I know, I didn't even notice.
Yeah,
And
it's so
then
quiet
Emma
in the
told me, and I
back.
was like, "What's
Prospective student checking it out. Yup. Yeah, not a problem. See you on